Hello guys, let's do section 1 of chapter 10. This whole chapter is about sexual reproduction, and we're going to discuss the process of meiosis or meiosis. Both are acceptable pronunciations, so let's dive right in. Our 3 essential questions. How does the reduction in chromosome number occur during meiosis? What are the stages of meiosis, and what is the importance of meiosis in providing genetic variation? So hopefully by the end of this video you can answer those 3 things. Here is the vocabulary that you should have gotten in class today. If you have not yet defined these terms, take a moment, pause the video, go write the definitions of these words, because if you know the definition of these words, the notes will be that much easier and you will understand it much more. So let's dive right in. Chromosomes and chromosome number. Characteristics such as hair color, eye color, the ability to roll your tongue, whether you have a hitchhiker's thumb, those are all called traits. The instructions for each trait are located on chromosomes, which are located in the nucleus of cells. The DNA inside the nucleus are organized within those chromosomes in segments called genes that control the production of a specific protein. Those proteins are what we can see and what give us our traits. Each chromosome in the nucleus contains hundreds of genes. So you have lots and lots of genes inside your cells. Now, here's a term that's going to look kind of weird to you guys. It's pronounced homologous chromosomes. Homo meaning same, logos from the, I believe it's Greek, meaning location. Okay, so this means that there are chromosomes that have the same location for genes. In humans, we have 46 chromosomes, but they can be arranged into 23 pairs because each chromosome has a mate, or one that has the same genes in the same location. You get one from each of your parents. You get one from mom and one from dad. The chromosomes that make up the pairs are called homologous chromosomes, and they are the same length. Their centromere is in the same spot, the same position, and they carry genes for the same traits. Haploid and diploid cells. To maintain the same number of chromosomes from generation to generation, organisms produce something called gametes, or sex cells, that have half the number of chromosomes when compared to a normal body cell. That is so that when mom and dad get together, okay, each one of them contributes half of their DNA to the offspring. Now, if they contributed their full amount of DNA, the amount of DNA in each nucleus would double each generation. So if we started out with 23 in generation 1, we would end up with 46. In generation 2, we would end up with 96. In generation 3, nope, I did that math wrong. But anyway, you guys get the picture, all right? The number would double with each consecutive generation. Okay, and that's not what we want. We want to have the same number of chromosomes in each generation, in each organism of the same species. Okay, so when these gametes are joined, okay, we end up with the correct number of chromosomes. The symbol N can be used to represent the number of chromosomes in a gamete. A cell with N chromosomes is called haploid, and a cell that contains 2 N chromosomes is called a diploid or diploid cell. Both pronunciations are correct. Meiosis is a type of cell division that reduces the number of chromosomes in a cell and produces gametes. It involves 2 consecutive cell divisions, meaning one right after the other, and they are divided into meiosis 1 and meiosis 2. So here we have a female, here we have a male. They each undergo meiosis. The female produces an egg or an ovum. the male produces a sperm. Each one of them have N number of chromosomes, which can indicate any number. In humans, it represents the number 23. N just means one set of chromosomes. When the sperm and egg get together in fertilization, you add the N's together and you get a zygote, which will develop into a fetus that has 2 N chromosomes. It has 2 complete sets. So, meiosis has the same phases as mitosis, which is why we're doing it right after mitosis. We start out with interphase, when the DNA is replicated and the chromatin begins to condense into chromosomes. We then have prophase 1. Okay, we're going to have a second prophase later on. Prophase 1, this is when the homologous chromosomes pair up. So the chromosomes get together with the chromosome that is the same length, has the same genes, and the centrosome is in the same spot. Each one of those chromosomes consists of 2 sister chromatids. So homologous chromosomes, meaning a pair of 2, has a total of 4 sister chromatids in it. As homologous chromosomes condense, they are bound together in a process called synapsis, which allows for something called crossing over. Crossing over is when chromosomal segments are exchanged between the pairs of homologous chromosomes. So they line up next to each other. See here, they're next to each other. Here, they connect, and wherever they touch, they will trade segments of DNA. And the end product, they are no longer identical to how they were when they started over here. This blue one could represent Dad's set of chromosomes, the 2 sister chromatids, and the red could represent Mom. Well, now we have a chromosome that is mostly Dad, but has a little bit of Mom. And we have one that's mostly Mom, but has a little bit of Dad too. Then, in metaphase 1, the homologous chromosomes line up in the middle of the cell. Okay, and you notice that they are lined up together as a pair, 2 by 2. So we have one on this side, one on this side. In anaphase 1, they separate, and you'll notice that it is an entire chromosome moving to either side. The homologous chromosomes separate and move to opposite poles of the cell. The chromosome number is reduced from 2N to N when the homologous chromosomes separate. Okay, because the chromosomes are still replicated, they're still doubled, but only the ones that are mostly Dad, the one that's mostly Dad moves this way, the one that's mostly Mom moves this way. So it's no longer 46 different chromosomes, okay? Now it's only 23. And then in telophase 1, it's pretty much the same thing as regular telophase in mitosis. They reach the poles, the new nucleus forms, and cytokinesis occurs. That's when the cytoplasm splits. Then we move into what's called prophase 2, which is very similar to mitosis. A second set of phases begins as the spindle apparatus forms once again, and the chromosomes condense. They then line up in the middle. They line up at the equator. Okay, meiosis 2 involves a haploid number of chromosomes. And then they separate in anaphase 2, just like they do in anaphase of mitosis. The sister chromatids are pulled apart by the centromere, pulled apart at the centromere by the spindles, and they move toward opposite poles of the cell. And then telophase 2 occurs, much the same way that it did in telophase 1 and regular telophase. The chromosomes reach the poles, and the nuclear membrane starts to form around that genetic information, and the nuclei reform. Then cytokinesis occurs and results in 4 haploid cells, each with N number of chromosomes. You'll notice that each one of these looks a little bit different. Okay, each cell has slightly different DNA than the others. That is what we want. That is the goal of meiosis. The importance of meiosis. Mitosis consists of one cell division that produces identical cells, which is what we want in our normal body cells. However, meiosis consists of two cell divisions that produce haploid daughter cells that are not genetically identical, which results in genetic variation, which is what we want. We want genetic variation because it gives the offspring a chance to have a better ability to survive. They might be taller, they might be stronger, they might be faster, they might be better at getting food. Those genetic variations might make that easier. Meiosis provides variation. During prophase 1, the chromosomes line up randomly at the equator. Okay? It doesn't matter which way the chromosomes go. Okay? We could get one from mom and one from dad on this side. We could get both from dad, or we could get both from mom. Okay? It doesn't matter. It is completely random which side of the cell the chromosomes end up on. Also, gametes at the end up with different combinations due to that crossing over, that trading of genetic information. Genetic variation is also produced when during crossing over and during fertilization, when gametes randomly combine. Fertilization, we get some chromosomes from mom and some chromosomes from dad, and we restore the regular number of chromosomes in that cell. So now we're going to compare sexual reproduction to asexual reproduction. Asexual, A means without, sexual means with sex. In asexual reproduction, the organisms inherit all of its chromosomes from one single parent. And the new individual is genetically identical to the parent, which might be good if the environment is very stable and it's not changing at all. That could be very bad if the environment is changing, because they're going to have the exact same traits as their parent, and they might not be able to survive. They might not be able to get food or water or shelter or escape predators. Sexual reproduction is beneficial because you get genetic variability. The rate of beneficial changes is a lot faster than in asexual reproduction, and beneficial genes multiply faster over time than they do for asexual organisms. And that's it, guys. If you have any questions, let me know. Also, guys, if you would like 5 bonus points, email Ferris with your favorite color, okay? So that I know that you have actually been listening to the videos. So, 5 bonus points. Send me an email with your favorite color. That's all.